Is it visible now?
Yes.
Hmm. Yes, Deepika, बोलो क्या? What is your question?
सर मैं ये कह रही थी कि फाइनल सर्टिफिकेट के लिए जो एग्जामिनेशन होगा फॉरेन एग्जाम और असाइनमेंट्स बस यही कंसिडर होंगे या ये फीडबैक असाइनमेंट सोल्यूशन भी यूनिट वाइज प्रोग्रेस लेसन प्रोग्रेस लेक्चर नोट्स ये सब हमने किया नहीं किया ये भी जज होगा।
तुम्हारे वहाँ पे इको हो रहा है। नहीं, एग्जाम के लिए वही काउंट होगा जो तुम लोग उस समय एग्जाम में आंसर्स दिखोगे।
और असाइनमेंट सर
असाइनमेंट्स काउंट नहीं होंगे उसमें।
नहीं होंगे।
नहीं नहीं, बट ये कोर्स कंप्लीट करने के लिए जरूरी है असाइनमेंट सबमिट करना, बट एग्जाम के लिए अलग पेपर होगा।
बस वही डिटरमाइन करेगा कितने मार्क्स आते हैं।
हाँ हाँ, वही डिटरमाइन करेगा। तो अगर किसी को इसमें अच्छा कुछ नहीं हुआ है या गलत हुआ है तो नो इश्यूज, डोंट वरी अबाउट इट।
सर
सो
मैंने तो हर एक ऑनलाइन सेशन अटेंड कर रही हूँ मैंने हर एक लाइव सेशन अटेंड किया है और वहाँ पे कोर्स प्रोग्रेस में मेरा 39% दिखा रहा है।
नहीं, इसमें इसमें ये ऑप्शंस तो है कि मार्क्स तो उसके रहेंगे, बट ओवरऑल तुम्हारी सर्टिफिकेट और वो जब मिलेगा उसमें मे बी ये सेटिस्फैक्ट्री नॉट सेटिस्फैक्ट्री में इस सब का रेस्पॉन्स, इस सब का कुछ रोल तो होगा। बट ओवरऑल मार्क्स जो तुम्हारे होंगे वो उसमें होंगे, जो तुम्हारा सर्टिफिकेट मिलेगा उसमें मेंशन शायद होगा कि हाउ एक्टिव यू हैव बीन
सर तो फिर
असाइनमेंट्स में।
उसकी वजह से कुछ प्रॉब्लम हो सकती है सर्टिफिकेट मिलने में।
नहीं देखो, असाइनमेंट्स अगर तुम करोगे नहीं ये
नहीं
सब कर,
असाइनमेंट तो कर रहे हैं सर।
तो फिर
13 असाइनमेंट्स कर लिए सबमिट कर दिए हैं वैल्यूएट भी हो गए।
हाँ, तो ठीक है ना, इसका भी रोल होगा। बट देखो, ये सब असाइनमेंट्स तो जो एग्जाम के लिए ऑप्ट भी नहीं करेंगे उनको भी असाइनमेंट्स करने ही करने हैं।
सर असाइनमेंट्स तो कर ली है मैं उसके अलावा पूछ रही हूँ आपसे जो फीडबैक का फॉर्म है या
नहीं
असाइनमेंट
नहीं, वो
सोल्यूशन हमने देखा है नहीं देखा है या यूनिट वाइज प्रोग्रेस लेसन प्रोग्रेस आ रही है या लेक्चर नोट्स ऐसे किए नहीं किए
देखो
ये
वो
पूछ।
देखो ये सब चीजें, फॉर एग्जांपल फीडबैक फॉर्म देना, ये सब अटेंड करना, ये सब दिस इज गुड दैट यू, इफ यू आर अटेंडिंग समथिंग यू हैव टू गिव द फीडबैक। सो जब तुम फीडबैक दे दोगे उससे तभी उसी पता चलेगा अगली बार कुछ इम्प्रूव करना है कुछ नहीं इम्प्रूव करना है, ये उसके लिए है। बाकी लेक्चर्स वेक्चर्स ये सब अटेंड करने हैं, ये तो ज़रूरी है क्योंकि हम भी अटेंडेंस दे देते हैं कि कितने लोगों ने क्या अटेंड किया है।
सर वो
तो
तो मैं कर रही हूँ
तो
एवरी
ठीक है,
लेक्चर
सो यू
एवरी
डोंट हैव टू
प्रोग्राम।
वरी, यू डोंट हैव टू वरी अबाउट इट। तुम एग्जाम के लिए ऑप्ट कर रही हो ना?
यस सर।
हाँ, सो डोंट वरी। सो उसमें सही होगा, उसमें ऐसे ही होगा जैसे अभी तुमने
तो
असाइनमेंट्स थे,
उसमें
कुछ
जो
डिफिकल्ट
कम
नहीं हो।
परसेंट आ रहा है 39% उससे तो कोई फर्क नहीं पड़ेगा ना।
नहीं नहीं नहीं, कुछ नहीं पड़ेगा उससे फर्क।
ओके ओके
हम्म,
थैंक यू सर
हम्म।
थैंक यू सो मच।
चलो, वी कैन स्टार्ट। सो दिस इज द fourth week lecture, and this is the last lecture of this. Maybe we have another lecture, but this is the last assignment section, my assignment this session. So after this, maybe we have 1 hour, another 1 hour session. So I will let you know later about it. Maybe you will receive the communication from NPTEL about it if it is there. So I will introduce myself again. Maybe some of new joiners are there. So my name is Ubaid Tariq. I'm a PMRF scholar at BSB Department of IIT Kanpur, and I'm taking this NPTEL live session of Introduction to Professional Scientific Communication every Saturday, 7 p.m. to 9 p.m. So we have from last 3 lectures we have been through, for example, how to write the paper, what types of scientific communication, uh, different parts of the paper, what is the sequence of writing. We have studied how to write abstract, how to write methodology and other parts. Now today we will move to the results part. So so in the result section, simply you have heard, I told you about, first you will write the methodology. After writing the methodology, because you know what experiments you have done, so you know everything about the methodology because at the time of doing the experiments you have noted everything, you have all the details. You just have to write that in the past tense that I already told you earlier. So after writing the methodology section, now results you only write when you got some results, when you have done some analysis. So so after that you will write the result section. But you have also you will also know what are the results, because when after getting the raw results, you will now analyze your results, and after that, after analyzing and interpreting them, then you will just simply write what are your results in the result section. You don't have to give that much background, or you don't have to discuss your results, or you don't have to give implications and interpretations, or some conclusions. You don't have to give that. You just have to write what are the results simply. So in the result section, simply state what you found, but do not interpret the results or discuss their implications. So you don't have to do that. As in the material method section, use subheadings to separate the results of different experiments. So in the material and material method section, I have shown you some papers also. So in material methods, you divide some experiments, and these experiments on the base of priority or on the base of flow, the paper you want, the flow you want to put your paper in. So you use those subheadings step by step. So same subheadings use the result in the result section. So that the readers, it will be easy for them to understand. For example, they have the interest in some particular methodology. They will look in that methodology only and then check the results of that methodology. And from these headings, it will be easy for them to look into that. So result should be present in the logical order in general. This will be in order of importance, not necessarily the order of experiments you performed. For example, you have performed different, some experiments you may have performed at start, and some experiment you may have performed at the end. But you don't have to uh, put those in that sequence. But you have to decide which one is important, or you have to make the story out of it. So to make the story and going from one step to other step, you have to uh, put your results in the end sequence only. Yeah? And in last lecture, I also told you you don't you are not going to put the single result as a one figure, but you are making the panel of the results. So you also have to analyze which figure or which result will go in this panel and another panel. There should be some correlation among these. So in result section also, you are going to describe it in a past tense. So however, refer the figures and tables in the present tense. For example, you are writing some this result. So this expression was significantly increased. It was found to be significantly increased as compared to control group. This was the example. But if you are referring that, because if you are writing something, it was increased, but you have to show that in the figure also, in the data also. So in the figure panel, if you refer that, but that refer line, so because it is in the present, you are referring that. So a result is in the past. You have done that means you have to, that's why you have to write result in the past. But if you are referring to the figure in the paper or table, that sentence you have to write in the present. So in the result section, both present and past will be mixed. Do not duplicate data among the figures, tables, a common mistake. So do not duplicate the data. For example, do not duplicate the data in the figures. Use the same data in one panel and another panel. So you have to make the clear uh, data should not be duplicated. Instead, use the text to summarize the you don't just if you are making any table and you are making any figure. So, for example, table has some data, for example, values, and figure has some data. So, table can be also interpreted and made in the form of figure. So, for example, schematic or some bar graph. So, but if you are making some table and giving all the information, you don't need to duplicate and use the same information and make the figure out of it. Either use either one of them, or figure or table. In some case, if you want to just give overview, so in table you can give the overview, for example, gene expression or something else. So you just give the overview that this is the level between the two groups. But in the table you give details of everything. So in case of those data which have large number of values, large n is large, so there are 10, 15 values that cannot be represented in a figure. In that case, you can make a table out of it. And table also is easy for a person to understand as compared to text, and figure is more much more easy to understand as compared to all of this. So you have to do statistical analysis also, because if you do any when you result analyze any when you interpret any result. So what I have done. Wait. So I'm here. So when you are making the figures, so you will also analyze those figures. So after you analyze it, so you have to give the significance. For example, in one group, in two groups, is there any significant change? So p-values are usually given. So in the text, you will write these exact p-values. What was the p-value, or was it less than 0.05, 0.01, 0.001? But in the figures, you will give the stars in this. I will show you in some papers and in the future slides also how to do this. So in the result section, what you should include an introductory content to understand the result. For example, 2 to 3 lines of about every result, some introduction, some introductory why you have done it, to understand why you have done it, and then you include the exact uh, what was the result, what you was shown in the result, what you got from the result. A systemic description of your result, highlighting for the teacher observation that are most relevant to the topic under investigation. So these are all the things, and after that you should when you write, for example, for some experiment, you're writing the result, so you should give one introductory line, then write about these results. In a panel there are 2 to 3 results, maybe 2 to 3 results you have to write about that, and at the end you can just give a short paragraph to conclude the result section by writing some overall from 2 3 results, what was the finding. You don't have to interpret, but just write overall. For example, overall it was analyzed that this happened, but this is like that, huh? You have to write it like this. So first some introduction part, then result, directly explain what was in the result, and then this conclusion. I will show some of the examples to you, so it will be easy for you to understand. So an introductory content. For example, this is one of the example. So they have done some of the experiment, but this is the introductory line that they have used. Since DRP is a key regulator involving in the mitochondrial fragmentation and is known as the GTPase that control mitochondrial fission. So this is the introductory line. They have also referenced this. If you are making any statement which you have not done, for example, this was earlier known, so you have to reference in the result section also, and in the methodology also, any section if you are using any line which you have not done. So now we check the it for the cellular levels of DRP and alter. Now this is what they have done, and then the author will write about it. But this is the introductory line that I was talking about. Before your result, you have to give this introductory line. So then inclusion of non-textual elements such as figures, charts. So in the result while writing, so in between you also incorporate these figures. If you have seen papers, so in papers there is no separate section of these figures. It is in between the text. So one panel is in between 4 to 5 subheadings. So though the subheadings are related to this, then there is some write up also. Then 4 to 5 subheading. Then there is a second panel. So this usually these non-textual contents figures chart they are in between this text. So here this is continuation of the same example. So this was the introductory line that I showed you. Now this is when you have shown the figure. So how to write this? As shown in figure 3, TRP level was significantly higher in brain tissues of Leforin deficient mice and Malin deficient mice. So they have included the figure. This figure would have been in the panel, not the separate. So it will be in the panel, but you in the result section you have to write this as shown in the figure 3a. So here in the bottom, the three would be written, and this part will be a. So I will show in the I have some examples, so it will be easy for you to understand later. Now, systemic description of your results. So you have shown these are results. Now you have to describe these results, highlighting the observations, some of the observations, what you have. You don't have to interpret, but you just have to write your observations. What are the results? So we have utilized, this is the same example following the same paper. So we have utilized following 4 different cellular models to test if loss of Loforin and Malin would result in mitochondrial fragmentation. So they have done 4 models. So just they have written like directly 1 model, 2 model, 3 model, 4 model. So they have given the information. So you have to describe in a systemic manner how after giving the introductory section, after giving describing the figure, then in figure this was exact observation. So readers, what they will do, they will after checking, they will check those figure also, and they will also check what you have written about it, what you have observed about it. So it will be easy for them to understand. The page length of a research section is guided by the amount and types of data reported. However, the focus only on the findings that are important and related to addressing the research problem. So I have shown you earlier. This time I will also show you some of the papers. For example, if I go to any paper, so if I go to this paper, so here this is the figure section, and these figures are also here in this journal. They have given separate also, but actually in the paper they are like this. So this is the figure 1. So it is in between the text. So this is was the result 1 figure is there. Result 2, in result 2 there is also the figure and in text it is these figures are cited that this is the figure, this is the figure. So depending on the journal, every journal has some policies that they will only allow some amount of figures. For example, they will allow only 4 panels, 5 panels, 6 panels. But if you have more than that, you have more than when you compile all the data, you have 10 panels and paper is only allowing 6 panels or 4 panels, whatever is it, for example, 6 panels. Then what you can do, you have to keep some of the data in a supplementary file. You have to, so here you can see that in this paper there are 8 panels, but you can see at some places, for example, it is cited that figure 3a. So this is this figure. At some places it is figure S. That means this is the supplementary. Like figure S1. So this is in the supplementary file. So you have to decide which one is important for you. If those which are important for you and which are making the story, those figures you have to keep in the main manuscript, and those which you feel that these are not that much important, and those you keep as extra files, and these extra files will be in the supplementary. So if you go into click on this link, figure S1, so this is the supplementary file of this paper, supporting information. So it has also some of the things, some of the materials and methods you have not written there. So you will write here if there is a space, because the paper journals also have this space limit issue. So these supplementary files will be helpful for you. So here you can see the figures are included. For example, that figure S1, it is here. So if you want to see that, so you can check the supplementary file. So you have to decide which figure is important, which is making your story. So on the basis of that, you have to choose the figures, and you have to keep them in a paper, and others they can be put in the supplementary file. So you have to focus on your findings, which are the important findings addressing your research problem, and that only you have to keep in the main manuscript. Now, one of the important part of that I already told you is at the end. At the end of the result section, for example, every result you have to write a short paragraph that concludes the result section. So here, from the example that I was talking to you about DRP1, so it can be like it has this paper he has written that taken together. That means all together the panel results what they has observed. Taken together, these observations support the notion that the increase in level of DRP could underline the mitochondrial fragmentation in low-4 in melanin deficient cells. So this is a concluding line. So I will show you this in the paper also. So if you go to the paper, if you go to the any results section, so this is the experimental section, that is materials and methods. Now if we are going to this result section, for example, characterization. So first at some points, this introductory line is not important, but at some points, introductory line is important if it's not that common. So first the figure is cited, then the first this NMR was performed, so they have cited the figure, then they have given some commentary about it. So after giving the commentary, they will at the end, I think here uh, there was not any uh, concluding line because all the results are different. But in those characterizations, I will show you. So like here, this paper they have not directly shown it, but after giving these figures, they have shown that the treatment of degradation products do not cause any significant effect on the cell proliferation. Overall, here they have explained it, the results, but overall 24 and 48 hours. Wow. These results showed that degradation products of power are not toxic. So this is that line that I was telling you. And before that, you are doing some of the lot of experiments. One, two, the figure one, 4D, figure 4C, and above there's a figure 4B. These all are about the cytotoxicity and degradation. So he has given some introductory line here: tissue injuring scaffolds degrade. This is the introductory line. Yeah, and after introductory line, I told you you have to give the reference because you are talking about some previous research. So this now why he has done to understand this in vitro degradation was done. So they have written that I have done this experiment. Then this experiment was mentioned that it is in the figure 4. Now figure 4 it is explained. figure 4 explanation done, then figure 4b, then this is the figure 4a, b, c, d, e, f. So d, e, f, all the things are written here. At the end, overall, from all these commentary, all these results, the overall concludatory line is this: these results showed that degradation products of PUAO are not toxic in nature. So overall conclusion was just this small line, but they have done a lot of experiments to check that. So this overall line gives the overall idea about what was overall message from these results. In the results section, simply state what you found. Do not interpret it. This I already told you. Do not interpret the result. This is another example. So leave it. So general rules about using the non-textual elements. Now I told you how to write the result section part, but you also have to include the non-textual elements in this. For example, figures. So non-textual element must have the caption consisting of the number and the title. So this, for example, this figure A, this is figure 2. Sorry, you don't have to only explain this figure in the result section. So you have explained it there, but in the figure below, you have to write the figure caption. This is called the figure caption. This, my figure 2, just between below the figure there. So they have written the mitochondrial fragmentation in the neural cell deficient for low foreign and myelin. So just then they have shown this is the A. So what is A? What is B? We have to write. So I will give you the example. Same paper if we go. So this is the figure 5. So this is figure 5. But they have written all the things about figure 5. You look here, 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 here. They have written all the things. But here, even if you write all the things, you still have to give the figure caption. In figure caption, you have to write what are these things in this figure. For example, if I zoom it, for there is absorbance, there are some other names written: TCP, PUAO, time, what was this, how this figure was, what this absorbance is correlating with time. All this you have to mention in that figure caption. So we'll go to the figure caption. You can see here we're looking to figure 5. The figure, what is figure 5? Overall one line. So figure 5 is a cell viability assay. So this is the overall about the figure 5. They have checked cell viability, how cells are viable or not, they are live or not. So A means this is A, C2, C12, the A is written there, is B is written there, and B, H9C2. So cell viability of C2C12, H9C2 on this PUAO films. So this they have written. So from this, if someone who is not able to understand directly from looking at the figure, actually in figure it is also there. You see, you can see it is also there. This blue color is TCP and this one is PUAO, but this C2C12 is also written there. Actually, sorry, this is blur, little bit blur, but all the things are written there. But if someone is not able to understand it, so the caption must be given. So caption is also very important. So A, what is in the A, what is after writing all about A, then B, then there is a C, D. So all the things, what is A, what is B, what is C. So all the things should be mentioned. So this is called the figure caption. Figure caption of So I already showed you in the paper. So this figure you have to this figure caption, the figure number you have that you have mentioned, you have to mention in the text also. So I will give you the example like this. So this is the result. So in the result you have to mention the result you have written, the text you have written, where it is shown in the figure. So table 1, figure supplementary A1, like figure 1A, figure 1B, figure. So all the things you have to mention. So every result you write in the result section, you have to mention in the brackets the figure where in the figure it is represented. So now some important rules about using non-textual elements in the research. So you have to follow some ethical guidelines. So what are these ethical guidelines? I will show some of the examples where you have to follow the ethics. So where are those ethics? So they are here. First, we will ethics are more about actually the figures. So we will first go right. I will first will tell you about figures and tables that are in the paper, and then I will show you what are some ethical concerns and ethical issues that you must be knowing. You should follow these. So, tips for an effective use of tables and figures in a research paper. First, check out what your target journals have to say on the issue. Some journals limit the number of tables that I already told you. Limit the number of tables or figures, and also have the specific guidelines, design aspect of the table. So I already told you, I already showed you in the last lecture. I will show you again some of the journals. They have guide they have guidelines about how to write every part of the paper. So in this section about journal publish with, if you click in publish with us, so for authors, so these are the guidelines about everything. So, so everything is mentioned here in these guidelines. How to write the paper, how to this method section is how to write the method section. So everything, even every technique, how to, if you have done some microscopy, how to label that. So links they have given to microscopy sections, how should be labeled, how figures should be made. So if you are targeting any journal, you directly have to, you must have some idea how I have to make the figures, I have to give this statistical analysis. So first decide that, and on the base of that, you can make your figures. So you should be aware of these specific guidelines. So you should be aware in which journal I am communicating this paper. So you have to make your figures and schematics on the basis of those guidelines. Next, decide whether to use tables or figures or text to put across the key information. So I already told you, if you have, for example, the data points, for example, compression test you are doing in the compression test. So whatever the force you are applying, so at every force there is some value of strain. So there will be a lot of values, thousand values. So those thousand values you cannot write in a table, because no one will be able to understand it or read it. So you have to make the overall conclusion of this. So in overall, to overall conclude the result, you just have to make the figure. But you have some data, for example, temperature, change in temperature, and gelation properties of the material, or melting properties of the material, whatever. So you will just write 2 to 4 temperatures, and then you will write what were the properties. So in the table. So you have to decide, you want to make the table or you want to make the figures, whatever you have to choose. After you decide to use the display items, choose the display item that best fits the purpose based on what readers focus. So all the time when you are writing the manuscript, you are making the figures, tables, all the time you have to think about the readers. You don't have to think about you, how I can write it, how I can uh, make it. You have to think about readers. If I represent it like this figure, will readers be able to understand it? Because you will not be able, when other persons will check your figures, read your paper, check your schematics, you will not be able to explain them on what thought I have made these. So it should be self-explanatory, and it should be easy to understand. Just ma by looking, the reader should be able to understand, but because figures are uh, the way of communication, to make the communication easy, to make the paper more attractive. Just at looking, they should be able to visualize it. So tables, you know that you have the numerical values. If you have some specific data and you don't know how to write that, and some data, it is if it is that if it is written, so it is not that much easy to understand, and that much easier to follow. So in that case, you can make the tables. In tables, you can put numerical values. You can compare and contrast between the data and between the characteristics. You can show the presence and absence of some specific characteristics in the table format. The best way to decide on the format of the table is to review a number of papers and see the format of most useful to us. So So it is, I already told you that you have to check a lot of papers, you have to check the journal guidelines. So on the basis of that, you have to make your tables, figures, and also you have to check how other people are doing it. If you are writing your first paper, how other people are making the tables, figures, and like that, you have to follow and you have to do like this, those papers. So tables just the numerical values, but in the figures, you show the trend, pattern, and relationship between the data sets. For example, if you are inducing some disease in an animal, so one group will be only with no disease, one will be with disease, and one will be in which there was a disease and you are giving the treatment. Now you are analyzing some out of it, you are analyzing your data, some parameters. So every time, you compare between the good control treatment, you compare. So you just interpret the trends. You don't have to give all the values. So you interpret the patterns, trends, and relationships between these two groups, between data and generally, and these patterns you can just draw in the form of graphs and data plots. You can summarize the research results, for example, in the form of graphs, data plots, pie charts. So present the visual explanation of sequence of events or procedures. So some of the sequence of events, so you can make schematics out of this, schematics, image, photographs, whatever. So some of the examples of figures are here. So this is one of the example. So if you have done the gene expression, for you want to do the gene expression, so you have different types of groups. For instance, set one was this PGM, GFP. So this is one group. This is the first sample, second sample, third sample, fourth sample. So you have 5 samples. In the 5 samples, you have to analyze different genes. So this is gene one, gene two, gene three, gene four. And in for every gene, for example, this is gene 1 A S and S, so this is the gene. So you have to check what was the expression, gene expression in between these different samples, five samples, so 1, 2, 3, 4, 5. So the easy way to represent is this bar. You cannot write the fold, for example, gene A name and fold. You cannot write this fold change. So it will not be. You can write it, but it will not be easy for a reader to visualize it and to understand it. But here, if you just skip this part, this x-axis, but just the reader can look at it, so he can out of it he can visualize that this has more expression, this is less, this is more than this is more than this. So it is easy for the reader to visualize and easiest way to explain. So if I will directly go to this figure and will not read the text of the paper. So from this I can easily understand whatever this gene. So this group has more expression than this group has less. This group has there it was increased. Just I may have to refer for these abbreviations. So these abbreviations will still be written in the caption just below this figure. It will be written in the caption of the. So it will be easy for me to just look into the figure. So figures and tables are often the quickest way to communicate large amount of complex data. Even if I write this, so this will be the very large paragraph. I will write gene one, I write about all these samples, how it was changed. I have to write the values. It will be very large paragraph. But the easiest way is just I am looking at it, I am able to understand it. So it is this is reader friendly. So that's why you have to make your data like in a way that readers can easily understand it. So you make the figures, then show to some layman or someone else, so he should be able to understand it. Not the layman exactly, but the one who has not made the figure. So you, if he understands it, so that means all the readers can understand it. So many readers will only look at the display items without reading the main text. So this is the main. Usually, if you also or search some papers, you can most of times we only look for the figures. So, uh, uh, therefore, ensure your display items stand alone from the text and communicate clearly the most significant. I already told you that even if I don't have to, if I have to, the good figure is I should not. The good figure is I should not have to go to this text to understand it. I should be easily able to understand just looking at the figure. That's the good figure. So you have to make the figures like that. Display items are also important for attracting readers, to your work. Well designed and attractive display items will hold interest to the readers. So if you have good display items, good figures, so readers will be attracted to your manuscript and they will read the full manuscript. When they are attracted, you will get more citations on your manuscript. So these high quality display images give the professional appearance to your manuscript. So these are some of the examples of the figures. For example, this figure, this is not good. Can anyone tell me the problems in this figure? What are the potential problems that you can see? Anyone? Deepika, tell me what are the problems in this figure? Are you finding any?
So ये जो टाइम बथ वाला ऑप्शन है, कॉम कॉम सारे सारे क्या लिखा है इसमें, क्लियर नहीं है लाइंस, इसमें कुछ समझ नहीं आ रही है।
देखो एक तो the color combination is not good. Here you can see that these lines in the background they are not that much visible. So this is one problem. Another problem is here the image is blur, but still these x-axis, y-axis, these values are too small to read. And here these values, there is no marking here in this y-axis. So where is the 0, 1, 2, 3? So it will not be easy to visualize exactly. So here you can this micro, usually this micromolar, it is micro is like in U. This is the common mistake. So some of their are writing U instead of micrometer. So you have to write exact the micro. Here the there should be space in this between this KDA and 24. There should be the space and some. For example, this A is capital, so B should be also capital. So small. So this is not okay. And every figure is not represented by this alphabet for this letter A, B, C, D should be written, but it's not there. So these are all. So this figure 4, it should not be written here. It should be in the caption. In figure, we do not write exactly the figure. This is the figure. So here you can see that this figure 4 is removed. A, B, it should be like this. This there's a space in between this. So points are there. So now different colors are given. Background was removed. So now it will be we can easily visualize it, the difference between two different groups. These are different groups, and the size of this is also increased. So you have to focus on these things when you create the figures. Uh, at the time of making the figure, this looks good. Uh, this this x-axis, y-axis, they look good, but when you make the panel out of it, when you compile 4 to 5 figures, when you make this figure small, this becomes too much small. So you have to make it extra large as compared to this, so that when you make a panel, they will be you will be able to visualize it from the far, because you are making it small. So because of making them small, the size also reduces overall the picture. So you have to keep these things in consideration. So what is the problem in above and why the below it is correct? You can see this looks too clumsy. The font is too big and bold. So here there is no space between IL. Even so, so this is not good. You should yourself see that it should look aesthetic. It should look proper. There should be proper spacing. There should be proper gap between the two line spacing. Everything you should have to. This is not good. This is good. Here you can say that this is not that much visible. You don't have to use that these type of colors. So this is comparably much better than this. You see, usually when you plot these figures, when you get a raw data out of it, so in the instrument file it will be like this only. So you observe, if you have some of you have done some work, it will be like this. But you have to manually make it large, so make it like this. So usually what people do is just export that file from that instrument and directly, without changing these things directly. So keep them in the paper and publish in the paper. So it becomes very difficult for the editors to understand. So when you make the panels, when you compass these, so that at that time you have to just overall check these. Is everything clear in these? So at that time you can change. So one of the important issue when you make the presentations or you make the figure panels, so you have to make the original aspect ratio or original proportion of the figure. For example, this is the rat. So but this may be the rat or some cell image or some any other liver image. any other organ image. So if you have to drag it, you have to drag it from the corner. So if you drag it from here, from here, from here, so at the end, the aspect ratio of this image may change. So in that case, so this look like this. So if someone makes narrow from these sides and pulls here, so it looks like the lean mice. And here, if you pull out this, so it looks like the fatty mouse or fatty rat. But this is not the case. So, so you have to keep them in the mind. And actually, those journal editors and reviewers, they actually have the tools so that they will be able to know that this is wrong. So in that case, the paper will be rejected. For example, in no specific feature within the image can be enhanced. For example, someone wants to enhance some color in the image, some fluorescence, something like this. That is not permissible. So nothing specific can be. You can overall do some of the modifications in the image, just the increase the brightness, increase the contrast, but overall image you cannot manipulate any specific thing, remove any specific thing, or increase any specific thing, which gives indication that this is the result. And there are specific tools that because of that it will be easily able to recognize that this is the manipulated image. So there are very lot of ethical concerns. If reviewers are after publishing or before publishing, they can reject it and ban the you from publishing in that journal. Or there are other concerns that your paper will be retracted if it is published. So these are very important concerns. When you go into the PhD, you should be more cautious about these things. So sometimes by mistake, to make the figures look good, we do some of the modifications. We have to refrain from it, and up to the ethical limits, you should know that. So up to ethical limits, you can just do these changes. So ethics in image processing for publication, for example, these two gel images, if you remove this line inserted, so this can be visualized like that this is the single gel image run at a single time. But just they are joined. These two images are just joined, and they are made a single figure was made out of it. So anytime when you join the figures or join these elements, you have to put this block line here. So it represents that these elements are joined. These are not the single image. So you just have to explain that I have run into the two gels. So this is this. So readers will be not be confused, but they can interpret it as a different result because of in a single gel and in different gel. So you have to be clear whatever you have done. But some people think what why it's important to just give this they are running the two gels. So I'm giving the result. So this is the result, but no, you have to exactly mention what you have done, how you have done, where it why the samples run in a single gel or two gels. Everything you have to mention. So here you can see that, for example, this was the image, and some uh, image it you want to magnify some. If you so this is black. So if I magnify some part of it and put here in a magnified image, if I not put this border here or some line here, so for readers they may look that this is the single image and these are this is just the large particle. This is large. This is also not allowed. So every time when you mm, merge those images, put one image on another, so or use also you are also showing the magnified portion of some of the particles. So you have to just mark a line out of it so that it will be readers will be easily able to visualize it. And schematics, you maybe know you may have seen these schematics in your 10, 12, and in your graduation, different notebooks, different pathways, Krebs cycle, and glycolysis. There are these schematics. If you write this glycolysis or Krebs cycle, it will be very and and also very hard to remember and hard to visualize it. But because of these schematics, it is very easy. And there are some signs. For example, this radiation, this arrow means it is activating this, and this is activating this. And this blunt means this is inhibiting, this is inhibiting this. Sora Finab, this is inhibiting rankle. Uh, this is activating this. So these signs we have to remember. So this simple arrow means activating, and uh, this blunt one means this is inhibiting. So from these schematics, it is easy to be able to understand if you give the radiation, this will happen, and then this bone resorption, whatever they have shown, apoptosis will happen. So, so in the one side they have shown what are the harmful effects of radiation, and here they have shown some of the beneficial effects of their therapy. So this is the easiest way schematic. So readers will be just looking at it, they will be able to understand it, and they will be also remember it also. So for example, this is the major roots of protein clearance. If we write this will be the very large paragraph. So you have to write about the transcription, protein synthesis. But here you can just show that transcription, then protein synthesis happen, then modification of proteins, then in proteins will aggregate. So chaperone mediated autophagy will happen. Ubiquitination happen here in this step. So cellular function, these proteins will go into lysosomes. They fuse with lysosome, auto lysosome. So all these things will is easy to remember, easy to understand. And for the readers also, it's very much easy. If you write this paragraph, it will be boring to read it, and you will not be able to understand it properly. So this is all about schematics and in the result. This was a result section in which the schematics were there, figures were there, all are in the result section. I will show you on paper, maybe from different journal. For example, this journal. So this is the graphical abstract. This is also overall. If you are writing abstract, so people will usually read abstract and they will be able to understand. But if you give same graph this normal abstract, you will make a graphical abstract, it will be much easy for the reader to understand what you have done. So these are different figures that are included. So you can see here it's more clear here. For example, figure 2. Figure 2 is development and characterization of this scaffold. Right? Then A. In A, it's written what is this A. So A you can write, you can see that the representative same image showing these electrosupan, these fibers. So what is B? So what is second part in this? What is third part in the A? So everything is written in the caption section. So sometimes you may think that this we are writing all the things here, what is here, and we are also explaining the same thing. You can see here uh, the same thing will be explained in the figure also. You can see here. So this is the way that sometimes it is repetitive, but for the this is repetitive because for the readers it will be easy. Maybe some of them are just looking at the figure, but when they look at this picture, they are not able to understand from this just on looking at this picture. Then it's easy for them to go just in the caption section, check F. So here is F. So in F, what is it? So it will be easy. It will be easy. So if so it will be easy for them to understand. And for this F, there is also written in the result also, discussion also. Maybe they are not they will not read it. They don't have that much time. But here it is easy. So this was all about results. Have you any questions about result section? Anything you want to know? So this figure is called the figure panel. So here, for example, by layer guiding this patch development and characterization. So this is one subsection. For this subsection, there is one this figure panel. In this figure, all the characterization which is done for this is written there, and figure caption is also written there. So in between. Now, when we go below, so this is the second subsection of the result. So now we have written something. Then in between there is this panel, whole panel, in which all these things are there, all the figures. Then the figure caption is just below there. Then some another part. So always this figure is in between the subsections of the text of the results. So this is the third result. So this is the third subheading of the result. And first intro, this is the introductory line with the reference. Then we do have written data. So in data there are these figures, and these figures are cited also. That this I have written this statement, and this statement you can see in the figure 4. So now I have written this statement, and this statement you can see in for example figure 4b. So I have written this. This is figure 4c. So this is the way in which you have to write your results. Now this is just live example. So all the results are there in figure caption, everything in the figure caption. And one important thing was this statistical significance. So here in the this in the caption you will write what this 3 star means. Just you will write what this star means, what this NS means. And in this in the text you will write what was the p-value. So here if it is single star, there are no stars here. So we check, for example, if it is single star, that means p-value less than 0.05. For 2 stars, less than p-value 0.01. For 3 stars, p-value less than 0.001. I will write it here. So if it is single star, if it is single star, so p-value is less than 0.05. If it is double star, p-value is less than 0.01. So anyone tell me which one is more significant? Which of the data is more significant? Single star, 2 star, 3 star.
Single star.
Why?
फिर क्योंकि वो उसकी वैल्यू बड़ी है।
नहीं, यही तो कंफ्यूजन है। 0 सबसे ज्यादा सिग्निफिकेंट है जितने ज्यादा स्टार हो, फॉर एग्जाम्पल 3 स्टार या 4 स्टार, जो भी है। व्हाट दिस पी वैल्यू, इट मींस वेरिएशन कितना है, व्हाट इज द प्रोबेबिलिटी ऑफ वेरिएंस, नो। तो अगर ये वैल्यू बड़ा है तो दैट मीन्स में वेरिएशन रिजल्ट में बहुत ज्यादा है। अगर तुम कोई भी रिजल्ट जब करते हो ना, तो यू डू इन अ डुप्लीकेट्स ट्रिप्लीकेट्स। सो अगर उनमें बहुत सारा वेरिएशन है, so it will not be considered as a good result. So in that case, the significance of the result will be less. But अगर ये छोटा हो, for example 0.001, it's less than 0.001, that means तुम्हारा वेरिएशन अच्छा है, that means कम है, that means रिजल्ट बहुत अच्छा है, तो that means ज्यादा स्टार आएंगे. So in 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 figures you just have to show these stars. You can show that. So इस स्टार से पता चलता है ये कितना सिग्निफिकेंट है इससे। और ये जो एरो है ना, फ्रॉम दिस यू हैव टू शो दिस, हम किससे किसको कंपेयर कर रहे हैं। एज कम्पेयर टू दिस, इसके 3 स्टार है, दैट मीन्स ये बहुत अच्छा रिजल्ट है। एज कम्पेयर टू दिस और दिस, एक ही स्टार है, दैट मीन्स ये इसमें उतना रिजल्ट नहीं है, उतना अच्छा रिजल्ट नहीं है। और ये जो ये बार है ना, ये दैट मीन्स ये इतना वेरिएशन है इसमें। एक्चुअली ये 300 वैल्यू था इसको, बट 300 या तो 350 हो सकता है, कुछ वैल्यूज 350 तक थे, कुछ वैल्यूज 350 ऊपर 50 नीचे, जैसे बोलते हैं ना, फॉर एग्जाम्पल 10 रुपए दे दो, दो ऊपर दो नीचे। सो ये 300 था, 500 50 ऊपर 50 नीचे। तो कुछ वैल्यूज 350 थे, कुछ 300 थे, कुछ 250 थे, कुछ 270 थे, कुछ 200 330 थे। तो ओवरऑल द मीन जो इसका ये था ना, मीन जो था, जो एवरेज था वो 300 था, बट द वेरिएशन वाज देयर मोर। इफ द वेरिएशन इज मोर, सो सिग्निफिकेंस विल बी लेस। ठीक है ना, समझ आया ये। So every time you see the paper, you see the figure. So if you see more stars in any, that means that is a good result. That is more less variation. If there are less stars, so there is less significance. And single star is least significant. अगर सिंगल स्टार भी नहीं आया किसी को, दैट मीन्स दैट यू कैन नॉट क्लेम दैट रिजल्ट। यू कैन नॉट क्लेम कि मेरी, फॉर एग्जाम्पल अगर तुम यू हैव गिवन सम ट्रीटमेंट, तुम लोग you have given some treatment in the rat. So you have to see that in the rat body weight was changed or not. So तुम लोगों ने जब दिया treatment, then body weight was changed. So when you check these and compare from without treatment and with treatment, so in with treatment the body weight was reduced. So if that reduction was just सिंगल स्टार, दैट मीन्स हाँ ठीक है, कम हुआ है। डबल स्टार दैट मीन्स और अच्छा है, बहुत अच्छे से कम हुआ है। 3 स्टार मीन्स बहुत अच्छे से कम हुआ है ना, एज कम्पेयर टू कंट्रोल। बट अगर उसमें कोई स्टार नहीं लग पा रहा है, जब तुमने एनालिसिस किया देयर इज नो स्टार, दैट मीन्स यू कैन नॉट क्लेम द बॉडी वेट वाज रिड्यूस्ड। सो उसमें फिर सिग्निफिकेंट नहीं है वो रिजल्ट। ठीक है, सो दिस इज वेरी इंपॉर्टेंट। So we are done with this result part. We will go to the introduction part. I told you कि when you complete methodology, then result, then third part you have to write is the introduction section. Because in the introduction section now you know what were your results. Then on the basis of your results you will set a background. So when you set the background, introduction part, you are just setting a background and also focusing on your results, so that readers will be more attracted to your after reading your introduction, they have all the clarity about your results. So introduction section should provide readers with the background information needed to understand your study and the reasons why you conducted your experiments. The introduction section should answer the questions, what questions, problems you studied. So this is very important. You have to give background information, just basics of background information. Then you have to give information. Then write some lines about that. Give your problem statement. Why you have chosen the problem. What were the solutions that are already explored, that have already done. So you have to write all these things in introduction. So while writing the background, make sure to cite the citations. For example, uh, I already told you the citations, how to do the citations. I have not shown you, but citations are important. I told you you have to cite the paper. You have to cite if you are writing something that you have not done. You are reading from another paper. You are now writing in your paper, so you have to cite it. Means you have to mention it that I have taken from this paper. So citations should be well balanced. So according to your results, so you should have you have to give citations. But there are some papers also which have given the results which are opposite to your results. So if they are contradicting, so you have to also mention that. Because if you have to also mention that, but तुम लोग सोचोगे हम क्यों दे दे contradictory result इसमें contradictory क्यों लिखे अगर हमारा जो रिजल्ट है हम उसी के बारे में लिखेंगे बट you have to mention it contradictory and then you have to give reason why it should be contradictory. So even if you not mention if you will not mention the contradictory result or contradictory कोई literature जो पहले तुमसे contradict कर रहा है, so maybe the reader, reviewer या editor they will find it and they will now ask you why you have not written. In that case, there are chance of cancellation. But when you yourself give the clarification, yourself write that, it becomes encouraging for them to interpret it and accept your paper. उनको पता चल जाता है हाँ this is the case, this is the contradictory, but it is okay. Their results are good. They are statistically significant. They look authentic, and now we can accept it. And citation, you have to use the current. For example, if you are doing research this time, you have to focus more on past 10 years, past 5 years, last year in recently क्या research हुआ है. You don't have to cite 90s, 80s के paper that are very old. Some papers may be very important breakthrough that you may need to cite, but you have to focus on the new citations, new papers. So new paper, you have to read new papers, you have to then cite those new papers on here, and they should also be relevant. यानी कि this should not be like the case that you cite any paper that is not relevant to your, so it should be highly relevant to your study, highly relevant to your paper. So the introduction section should not be long. For example, different journals have different guidelines. You have to follow that. But briefly, it is just between 250 to 600 words. Depends on the journal. Some journals accept allow it for 600, some for 250. Yeah? So you have to check the journal guidelines and everything. And usually, pay, uh, these journals they do not keep limit on methodology or results. They want you to explain fully, explain all the things in that. But in introduction, they want you to be concise, to be focused. So in that, in the introduction, in the abstract, they keep the limit. So you have to follow these things. So this is the earlier slide in the introduction. What are the important things? The important things are what you have to write. You have to write some of the questions like why is your research important? You have to mention this thing. What is known about the topic? So from the literature, what is already known, you have to mention that. What is your hypothesis? After giving the background, then you have to follow it to your hypothesis. Now, this is my hypothesis. I want to do this. Then, what are your objectives? So, what are your objectives? What you want to do? Then you will go to methodology, research, and discussion section. So, these are all things I have shown you earlier. General is a strategy for writing an introduction. Establishing some of the important things is establishing an area by research, identifying the research niche, placing your research within the research niche. So you have to establish your research area. For example, research area highlight the important topics. So in which research area your research is coming. So when you highlight the research area, so in that research area, which type of in the research niche, small subheading or small research niche you have to identify, and on the base of that you have to choose. You have to write your introduction and you have to cite papers on related to that research niche. So you have to you should know intent of your study. You have to outline the characteristics of the study, importance of results. On the base of that, all you have to write your introduction. Now comes to the discussion section. As I already told you earlier that after writing, after methodology results, then you will go to introduction. In the introduction, you will now know that you have read a lot of papers, and in the discussion, what you have to do, you have to just correlate these papers with your results. So discussion is very important. You are correlating. So from introduction, it will also help, and from results, you already know that. So writing it after results and introduction is very much beneficial. So the purpose of a discussion section is to place your findings in the research context. For example, what is already there to explain the meaning of the findings and why they are important without appearing arrogant, and you don't have to appear arrogant. So you just have to mention, you don't have to mention like many है सब कुछ किया, this is very important and nothing else was done earlier. मतलब not like this, but you have to correlate. और तुम ये नहीं है कि you don't have to prove others wrong कि वो अच्छा वो that result was not good but our was good. No, you just have to correlate, but you can just mention if there was some contradictory result, you can just mention that our findings are not contradictory with these results, and then you can cite those also. In discussion also you have to cite because you are linking your results with the already published literature. So already published literature to which you are linking, you have to cite that. The structure of the first two moves is almost mirror reflection of one another. So this is just the uh, mirror reflection of the introductory section. So you have to write some parts of the introduction in the start. In the start, you just have to start like the introduction. So I will show you some of the paper just now. So first few lines is just like introduction. Then you are moving, focusing on your research, and then correlating these all these things in that. So moves in the research paper. What are the important things you have to keep in consideration while writing the research discussion? So you should keep in mind the study's major findings, recent context, and closing the paper. So first you will start with just giving some basic introductory line, just the reflection of the introduction. Then you start writing your major findings, state this study's major findings, explain the meaning, importance of these findings, consider alternative explanation of these findings. Then you will correlate with, compare and contrast your findings with those of the published literature. And this should be the recent literature. You have to give the focus that this literature should be recent. You have to explain any discrepancies or unexpected findings. If you have any contradictory findings, you have to mention that. You have to give reasons that. You have to state the limitations, weaknesses and assumptions of your study, whatever you assume. Maybe sometimes you have assumed something after after observing some results. So you assume that this may be the pathway, or this may be going like this. So you can first narrow down, then broaden it up at the end by summarizing the research question, indicating the importance of the work by stating applications, recommendations, and importance of your study. So after discussion, there is a conclusion. Your discussion and conclusion section should answer the question, what do your results mean? So discussion means what overall your results mean, and in the conclusion also there is a take-home message from your discussion. So problems you have to avoid. Do not waste time restating your results. So as you have already mentioned, what are your results? You have mentioned in the result section, you just have to listed your experiments. You don't have to just write those again here. But you have to just correlate. Do not introduce new results in the discussion section. So those results which you have already mentioned in the result section, write discussion about only those. You cannot just mention a new result here in the discussion section. So I have already shown you earlier different parts of the paper like acknowledgement, reference acknowledgement. You thank all the people who helped in the research but did not qualify for the authorship. Those who provide intellectual assistance, technical help, and special equipment or material, funding body, research grant, for research grant, they all are acknowledged in your paper references. So all the citations which you will do, you will cite the papers. Those you have to write in the reference section. So before going to the these questions, I will show you uh, one paper, the same paper. So this was a discussion. In some papers, the introduction and discussion is same, written in the same heading. So at the same time, they have explained the results, and at the same time, they have written the discussion. Some papers, result part is separate and discussion part is separate. That's why I already told you you have to keep in your mind in which journal you are submitting. For example, you just keep on writing the way you think, and then you are submitting in the journal which has uh, altogether different guidelines of writing. So you have to keep in your mind before writing in which I am journal I am targeting and write according to that. So here results and discussion is same. So at the same time results are mentioned and also discussed. So that's why you can see references in between. So conclusion, this is the conclusion, this is the overall conclusion of the paper. This is the authorship contribution, authorship contribution, how different authors how they what they have done. So what this author has done, what this author has done, all the things are mentioned here. So declaration of is there any conflict of interest? So this is a acknowledgement section. So here you can acknowledge different these research groups, different funding agencies that have given the funding, or someone if anyone have helped you directly uh, in your research, but these authors do not qualify for the authorship. So you have just have to acknowledge them here. So this is the reference section. So reference one, reference two, all the references that you cited will be here. So now uh, you may think how to reference these. One way is you manually write one and in the bottom write what is one, what is two, but is that is too hectic. It is not that much that easy to do referencing like this. So there are two tools which I will show you today. One is Mendeley, one is EndNote. So this time I am using Mendeley. Earlier I was using EndNote, but I have not EndNote installed in my laptop. So this time I will show you the Mendeley tool. This is the software. So I will open the Mendeley Reference Manager. So this is a software which used for this for referencing. So it is very much easy. So it easily manages your references. So you have to do nothing. You have to just make the account. This is the free software. You don't have to purchase it. You have to download this mainly from mainly reference manager from Google. So download the file, install it. So here will be the papers. But how to add the papers in this? So is this software also have the this version, online version? So in online version, so you can just can just search. No, no. So there are two options. One way is directly, for example, uh, you have installed this Mendeley. So one way is directly you click on the paper site. So you save this citation, for example, RIS. So this RIS was downloaded. So you can see here, I have just downloaded this RIS, but this is the reference. So now I will go into the Mendeley. So I will add a new reference.
What is RIS?
This is the format of, there are different formats. So RIS format you have to download for Mendeley only. There was one another format I will show you. So for example, this RIS, it works with EndNote and Mendeley both. So this is much more universal. So this is one BIBTEX. These are different formats of citation. So this RIS, it is compatible with EndNote. This Mendeley, this time I am using Mendeley. So you have to add new references. So if you have downloaded this EndNote, whatever you have downloaded, I have downloaded RIS. So I will add import library from RIS. So this was the reference. I will open it. So it will be just shown here. This was this is what I have added. This is one way of adding your references in this. So your references will be, I can segregate it on the different basis. For example, this paper one, paper two, what the I can segregate it. Another simple way of adding is just I will show. You will search mainly here, mainly reference. Here, this is my account. So same from the same account I have logged uh, in this software. For example, this is the account, my account, Uber. So in the same using the same account, I will log in the web. So this is my account. So any paper I will search here. For example, I want to cite this paper. No. So what I can do, this is the DOI that I already told you. Copy this DOI, or I can just copy paste this text title also. From this you can search also. But to be more accurate, I will search this DOI, just searching the paper. So this was the paper. So I can just read it, but I can directly add this to library. So now if so now it is in the mail. अब अगर मैं अब library में जाऊँगा ना, if I go in my library, you can see that this is directly added. You see, तुम्हें कुछ नहीं करना है, हो गया ऐड ये. अब इसको पेपर में कैसे डालते हैं? For example, this is my paper. तो मैं कहीं से कुछ टेक्स्ट लेता हूँ so that for example ये पेपर है. So this is the paper you are writing. तो अगर तुम ऐसे लिखने बैठोगे यहाँ पे ब्रैकेट करके one reference one, then here what is one reference, so it will be very much hectic for you. क्या कितना लिखोगे, how much you write, and maybe you have written one here and after some time you will incorporate reference above it. Now this one becomes two, so you have to change everything, every number. So this medley, it can be added also in the Word file also. So I have added it. So this is Mendeley site. So if you are not able to understand how to download Mendeley, how to add it in the Microsoft Office, so you can directly search some YouTube videos. So you will find how to do these things. It will be easy. So you just see अभी मैंने जो पेपर ऐड किया था extrasolar vesicles, so it is already there. ये सारे papers, all papers are there that were in my library. So you can just select this paper, this one, maybe four papers, whatever you want. So you can select in where you want to insert it citation. So like this, अगर यहाँ पे करना है तो if I want to insert it, तो यहाँ पे just cursor, keep your cursor here, then insert. You can see one or two were inserted here. ठीक है, ऐसे ही.
So what if we want to give text references in between
हाँ हाँ.
instead? So instead of numerical value for reference, if we want to give text references.
That can be also changed in the setting. I will show you. So here if you want to insert, so insert it here. If you want to insert this here, just select this, keep it like here. But you just have to add your references in this library. So when you add here, so from this you can insert any place. But you should be, well, you should know where I have to. So here also, if you want to insert this, so insert. So all these numbers. Now you can see that this was 1, 2, this is 5. If I insert some reference here above this 1, 2, यहाँ पे अगर मैं I will insert. For example, I will insert this. So देखो खुद ही ये change हो गया. This 1 plus 1 becomes 2, 3. This becomes 4, 5, 6, becomes automatically 6. So you don't have to worry about it. So now you are saying that if I want to some papers, they have policy that this 1, 2 is not there. It is author is written. So you have to change the here style of. So there are all the styles of all the journals are there. For example, Nature, update citation style. So Nature has this style, small, small, one, small, two. It will be automatically updated. So if you want to change it to this, so this is this. Some journals, you can see now it is right. This journal has a policy, American Psychological Association. So you just have to select the journal, and it will automatically change your references according to that journal. You can see that all these are now according to this journal. Huh? Now you have done it. When you click here, insert bibliography. So all the references uh, that you have incorporated, like I will, all the references that you have incorporated was all 1, 2, 3, 4, 5. In your paper, there will be 100 references maybe. So you don't have to write. All these things are incorporated by itself. You see, 1, 2, 3, 4, all the 6 that I have included. If I insert 100, the 100 will be written here automatically. And if I'll change it, the number, anything you can do anything. So this is that much easy. It makes your life hell easy because it is very difficult to manage references. Anyone have reference earlier, some of your paper or something else, or you are seeing this first time?
So I have tried to do some melody.
Which software?
No software, just by myself.
You What? It was a paper.
Yes, that we are doing.
How difficult? What is? Was it
It
to
was
reference?
difficult.
Too difficult because when you change the references, you remove one, so it is very much hectic. So you download this Mendeley software. This is free. EndNote is actually not free. I don't know. This is free for you or our institute has subscribed. I think our institute has subscribed to elsewhere. But you can check the graph this crack version of this EndNote. EndNote is same like this. The steps I have shown same EndNote, but there are different options there.
And note,
END. I will show you. So for EndNote, you will easily find the crack. This EndNote. This trial version is also there for 1 month maybe. So you can just install the trial version and check this. But but it will be only maybe 30 days. So if you are planning to try it in 30 days, something, so you can try it. Otherwise, you can look for a crack version of this EndNote. So I'm not encouraging to use the crack version, but can check. So it will be very helpful. So now we will move towards this. This is the referencing. This is very important because I told you everything about the paper, but you maybe these things you may be aware, but I know that most of these students they struggle with the reference. They don't know how to manage these softwares. So later on, if you need some any help, so you can contact me on my this YouTube channel. You can comment on this or something else, so I will help. You can comment on this lecture, so I will can share you the video link or anything else. So if you in future after when I complete this session, so if you need my help in any part, so you can contact me. But this is the one way to contact, huh? So I will be there. So now we'll move towards this solving this session. So some of the questions, first question is which of the following choices accurately describe the role of the journal editor who handle research paper submitted to the reviewer? Confidentiality, professionalism, and data protection. What do you think? Everyone should answer, not only Deepika. Yes? If no one is answering to Deepika, you can answer if you know. For others, they can write in the chat box.
professionalism, professionalism.
So why do you think confidentiality and data protection is not important? This is about the
Because
journal editor. When you submit something, some paper, it first goes to the journal editor. It's the head of the journal who is managing the journal. So professionalism, yes, he should have the professionalism, but he should also have the confidence. He should keep your results, your study confidential. He should also protect the data because you are giving all these things to some person. So he should protect. He should not leak your data, your findings. Maybe
yeah,
some other group is also doing the same type of research, and they are struggling with something, or they just want to publish it before. So he will just give them information that they have given. So this should not be the case. It is unethical. So he should mention the confidentiality, professionalism, and data protection. So all of these is the answer. The image given here represents an example of a figure prepared for a research paper. Here two images A and B connected with a dotted line or a square of and a square in figure A. Which of the following options accurately describe the most likely explanation for such representations? These two images A, B are the two different sources and the insert likely was drawn to highlight the similarity in the pattern of granular structure. The insert identifies the actual size of the object when printed in the journal. The image on the right side is likely a higher magnification viewer of the inset shown on the left image side. All of these, which one do you think? Deepika, which one do you think is the answer? Are you writing, anyone writing in the chat box? Actually, today I am using different browser, so it is difficult for me to track. So option three, yes, option three is the answer. You can directly unmute yourself and say it because I have to go back to that browser. So option three, actually, this always we show this dot square. That means this square is the area in which we have zoomed it out. So this is the zoomed version, magnified version of this image, magnified portion. So you must have seen in the different papers also this magnification is done this way. Writing a discussion section is the most challenging part of the research paper. Some of the pointers that are often used to draft the discussion section are given below, not necessarily the order of execution. Identify the option that correctly orders these steps. So in writing the discussion section, what we have to focus? So what is the correct order? Define the significance. These are M and OP. You have to tell me the correct order in which we have to write it. Define the significance of your discovery and explain its meaning. Contrast and compare the findings you obtained with those other research that has been published. Explain the significance of the work by discussing its application, recommendations, and implications. Describe the study's assumptions as well as its strengths and weaknesses.
Excuse me, sir.
Yes, किसी ने आंसर वहां से भी किया है बोलो.
So मैंने कह रही हूँ I'm having to go now. I have to leave. So आप कुछ और important अभी discuss करने वाले हैं क्या?
You have to leave, so you can leave. Huh?
आंसर वो मैं एक बार और कंफर्म कर दूं कि वो प्रोग्रेस यूनिट वाइज प्रोग्रेस रिव्यू का कोई मतलब तो नहीं है ना सर्टिफिकेशन से
Which progress? I'm
यूनिट
not.
वाइज प्रोग्रेस रिपोर्ट जो इसमें प्रोग्रेस अंडर आ रही है
हाँ हाँ उसमें कोई ये नहीं है.
उससे कोई मतलब नहीं सर्टिफिकेशन का
नहीं नहीं don't worry about that.
ओके थैंक यू सर
Okay.
I'll get in touch with you through your YouTube channel
हाँ हाँ you can get in touch with me. Yeah.
ओके थैंक यू सर
ठीक है thank you. So the answer is this. First MNOP. So this is the answer. So first we have to define the significance of our discovery and explain its meaning. Then we have to compare and contrast with other research. And after that we have to explain its significance, its applications, its implications. And at the end we have to draw some assumptions and mention its strengths and weaknesses. So this is the order of writing the discussion part. So fourth question is science journals frequently require authors to Clarify that the submitted manuscript is not the part of the simultaneous submission. Here, simultaneous submission refers to. So, when you submit any journal in a submit any sorry in a submit any paper in a journal, you have to follow some guidelines or you have to follow some rules. So, one of the rules is simultaneous submission. So, what this simultaneous submission refers to? I will directly told you. Uh, So these are the four authors, the same set of authors simultaneously submitting two or more papers, each describing different work. So no, you can, but this is not a simultaneous submission. You can submit if you have two works, you can submit at the same time in two different journals. Same set of authors simultaneously submitting the same work results to two or more. This is actually submit. This is not permissible. So if you have same, for example, if you have the paper, so you cannot submit directly at the same time in the two journals. So if you're submitting in the one, if that rejects, then you can submit in another. Sometimes the people think we will save the time, we'll submit in the two and think then decide maybe one of them rejects and one of them uh, but there is a possibility that one of them will accept it. But this is very unethical and that should not be the case. In some case, if it was found that your paper will be then not banned from these journals, it will not be then you cannot submit in these journals. Simultaneous submission is almost identical findings in to the same journal, but we never do so. But this is the answer. Two is the answer. Scientific journals often obtain publishing rights from authors before publishing the work of the authors in the journal. Which one of the following option is not true with regard to publishing rights transferred to the journal? So we have to give the publishing rights to the journal. So when you submit the journal, all the things review is done. Then you have to do at the end you have to do the proofread. So they will they will format the journal, your paper according to the journal journal these guidelines, and then you have to just check it, and at the end you have to give publishing rights to them. So which one of is not in true with regard to publishing rights transferred to the journal? Preventing the authors from republishing data and figures on commercial websites and books. This is the one option. So you can this is this is not true. You can even after publishing you can republish your figures in the websites or books. No? You cannot publish in the websites or books. Sorry, preventing the authors from republishing their data in the figures that thesis or lab reports. But in lab reports and thesis, you can put your data when you are publishing, preventing the authors from publicly sharing the in their website or so. This is also allowed. You can share in your website public articles or some public forms. In present, you can present it. So this is allowed. So this one is also allowed. You can use in the thesis or lab reports, preventing authors from claiming the copyright of the text or image. This is also because this is your image, you have the copyright. You can claim the copyright of the article, but this is not allowed. Preventing the authors from publishing data uh, and figures on commercial websites or books. The group of students created a slide presentation to explain gluten, a protein naturally found in some grains. Explain the slide to select the appropriate critique for the options provided below. So what critique you will do to this slide? Too dark background image. We do not add much information to the content. Too many colors in the text material. The color contrast of the font with the background is hard to read. बोलो क्या है? I think all of these are the issues. Too dark background image, so the image is given so and does this image doesn't give any information also, and these colors are in too many colors. They are green, some black, red, and color contrast is not, some text is not visible. So these all should not be. You have to check actually. You should not have to follow these things. So your image should be clear, your background should be clear, your text should be easily contrast, it should be visible as compared to background. Majority of the papers published in scientific journals contain the section titled Acknowledgements, in which the authors recognize the contribution of individuals or organizations. Which of the following statement is not ideally be included in the acknowledgement section? Which one? I just showed you this. The sponsor who funded the research project, yes, it should be included. The expert who read through the manuscript provided critical inputs, yet it should also be included in acknowledgement. A colleague who shared some research reagents, yes, it should also be included. A friend of author who helped him financially during the elementary scenario, this should not be included. This is not the direct contribution in this study, so this should not be included in the acknowledgement section. So following is a statement with two sections that are denoted in blue and red color fonts. Examine the statement and select the options that is true with regard to the guidelines followed in a research or paper. A research publication frequently adheres to the standard format for citations, and such formats ensure the ethical guidelines are adhered to correctly. So what is the answer? So which guidelines should be followed? A research publication frequently adheres to the standard format of citations. Such formats ensure the ethical guidelines are adhered correctly. So which statement among them is true with regard to guidelines followed in research? Both segments are correct, both segments are incorrect. Segment shown in the blue color is correct, and segment in the incorrect segment shown uh, in the blue color is incorrect. So a research publication frequently adheres to the standard format for citations. Yes, this is correct. Uh, and such formats ensure ethical guidelines, but there is no link between these. I think this blue is correct and the segment shown red is incorrect. There is no link. A format ensure the ethical guidelines are added correctly. This is just the format of the paper, so it will not ensure the ethical guidelines. The ethical guidelines are separate. Which of the following is not an effective method for displaying the overall trend of a large data set? Bar diagram, pie chart, table, and line diagram. So overall trend, bar diagram, in bar diagram I already told you, you are showing the overall trend. In pie chart, you are also showing the overall trend. You are not just showing all the results or the values, you are just showing the overall trend between two groups is increasing, decreasing, all the things you are showing. Line diagram, this is also the overall trend, but this tables is only one option in which there is not overall trend, it is just the exact values that you are mentioning. A group of students created a novel strain of an infectious fungus whose spores cause skin irritation when they come in contact with human skin. Students wished to determine if the newly developed virus was infectious. Based on your knowledge of research ethics, choose the committee that needs not to be approached for permission to conduct the proposed research. So, so these students they want to use some infectious fungus to conduct some study, So now to conduct any study, so on subjects, so you have to take the ethical clearance from that. So which ethical clearance you should take using this infectious strain? Institutional Ethics Committee for research involving human subjects, Institutional Animal Ethics Committee, Institutional Biosafety Committee. Which of none of these above is not the option because you have to take the approval. As this research is intended for the human, so this is important. Institutional Ethics Committee for research involving human subjects. You have to take, but Institutional Animal Ethics Committee, this is not important because this is concerning to animals. So this you don't have to take the permission. But as this is the infectious fungus, which has some biosafety concerns, you also have to take the permission of Institutional Biosafety Committee to conduct this research. So this was all over about this uh, week 4 problem solving session. So all these sessions are uploaded in my YouTube site. So you can just check this Ubaid Butt 55 or Ubaid Tariq Butt and you can follow this if you want to listen to the lectures later on. So this was all about today's session. Maybe we will have now one another session, one last session. So if you have any questions, you can ask. Only I think Varsha is here. You have any question, Varsha? So if there are no questions, we can conclude this session. And if we have one more session, we will get information from NPTEL. Thank you so much for joining.